Dear students, today we will talk about production of primary metabolites using microbial cells. So in this section we will be talking mostly about upstream engineering of the microbial cell as well as the production environment. So when we talk about upstream engineering, there are two components. One is strain improvement and the other is process engineering. Among the different ways of improving a strain, here I will take some examples, and among the examples the first one will be improvement of a strain by deregulation of pathway genes using analogs and increasing gene copy numbers. And for this I will be taking an example of lysine production. In the second example, I will be talking about strain improvement through a strategy of media chemical changing and different media chemicals which will affect or bring about the uh, improvement of the strain. So for this example, the production of citric acid in industries will be uh, discussed. The third strategy of strain improvement that I will talk about today is overexpression of genes, and for this example I will be taking penicillin production. The next example of strain improvement is constitutive expression of T7 RNA polymerase to increase transcription, and for this I will be taking GFP, green fluorescent protein production, as an example. There are other methods of strain improvement also. The next example is of inactivation of restriction modification system, that is in the case of amylase production. Similarly, one more strategy that is used in the case of production of polyhydroxyalkanoate is co-expression of pathway genes to increase the production. And in this section, the last example I will be talking about is of increasing copy number of the key genes involved in the production involved in producing a protein or enzyme which will bring about the bioprocess, which is biotransformation. So from this from this slide what we understand is that there are various methods or strategies of strain improvement, and some of these strain improvement strategies are also bringing about process engineering, and these two are clubbed together. For example, in the case of citric acid production, process engineering itself is bringing about strain improvement. That is, there is no separation between strain improvement and the process engineering in the case of citric acid production. So let us see all these strategies in detail one by one. The first example is of lysine production. Here in this slide, the production of lysine in Corynebacterium glutamicum is shown. In this microorganism, in this bacteria, the production of lysine is through a complex pathway, and the starting point of this pathway is aspartate, which is obtained or which is produced from pyruvate. So the aspartate is converted in the presence of aspartokinase or aspartate kinase into L-beta aspartyl phosphate, and this in turn is converted into aspartate semialdehyde in the presence of aspartate dehydrogenase enzyme, and from this aspartate semialdehyde in the presence of dihydro dipicolinate synthase, lysine is produced. This is not a one step process. There are many steps involved, hence the uh, line here is dotted, which means that there are more than one intermediate involved. So the lysine production from aspartate is uh, achieved with the help of these few key enzymes, which are encoded by various genes in the, like aspartokinase here AK is encoded by lysine, whereas aspartyl phosphate dehydrogenase is encoded by ASD gene, and the key enzyme which is involved in the case of production of aspartate semialdehyde is encoded by its corresponding gene, DHPS gene. Now this pathway is a branched pathway, which means that there are other primary metabolites also which are produced from this pathway or the branch of this pathway. For example, aspartate semialdehyde is involved in the production of homoserine, and the homoserine in turns branches out to produce threonine, which is also another amino acid. Then homoserine is again a branching point for the production of methionine involving various steps. So every biochemical pathway or metabolic pathway has many points of inhibition, and these inhibitions may be repression or feedback inhibition, feedback repression or feedback inhibition. Here in this pathway, lysine production is controlled by feedback inhibition of lysine feedback inhibition of aspartate kinase or the gene lyse by lysine and the threonine, and the threonine is also involved in feedback inhibition of the homoserine dehydrogenase gene. Apart from this, methionine is involved in feedback repression of homoserine dehydrogenase enzyme. So there are many points which are to be considered when we allow free and unregulated or deregulated production of lysine from this pathway. But other metabolites like threonine, methionine, these are also important metabolites in this bacteria, Corynebacterium glutamicum. So uh, in order to bring about accumulation of lysine or production of lysine in um, industrially acceptable level, what we need to do is we need to deregulate certain enzymes. Before this, let us see some more facts about lysine production in Corynebacterium glutamicum. Apart from feedback inhibition of aspartate kinase by lysine and threonine, there is low level of DHPase production, which is dihydro dipicolinate synthase. So if there is low level of this key enzyme production, then the accumulation of lysine will be not up to what we expect. The another fact about lysine production in this bacteria is low level of pyruvate carboxylase production. Yes, production. So pyruvate carboxylase is involved in the formation of aspartate from pyruvate. So if the starting point L-aspartate itself is in short supply, then there will be no possibility of high level of lysine production. Another fact is that the AK or aspartate kinase enzyme, which is the first important enzyme for production of lysine in the whole pathway, itself is produced in low quantity or in low level. So these are the facts that we need to overcome in order to overcome these biochemical barriers present in Corynebacterium glutamicum. What we need to do is we need to look for biotechnological means of getting over or overcoming these barriers. So here what we have uh, got after uh, after a lot of research is that there are some strains of Corynebacterium glutamicum which are mutants, those mutants which cannot produce homoserine dehydrogenase enzyme, because uh, gene which is the gene which is encoding homoserine dehydrogenase itself is absent in this mutant. Also, there are strains of lysine mutant and the pyruvate kinase gene mutants, but all of these mutants cannot be used. But one mutant that will be of use to us is that homoserine dehydrogenase. If a strain is deficient of this homoserine dehydrogenase encoding gene, then that strain will help us in accumulating lysine, because the branch point from aspartate semialdehyde to form threonine and the methionine will be cut off because of the absence of this enzyme. However, there are other possibilities. If there is no any mutant which is lacking in the homoserine gene, then all of these important genes can be somehow mutated in order to increase or decrease their um, production or their expression in the bacteria. So when their expression is regulated in such a way that fits our requirement, then there will be production of lysine. This is one way of bringing about enhanced lysine production in Corynebacterium glutamicum by inducing mutation in the key genes involved in the whole process of lysine production. Now, once we get all these mutations in place, suppose, then what we will do, we will have to engineer some other strategies for um, which can be coupled with this mutation strategy to bring about increased production of lysine. The strategy is of increasing lysine apart from this mutation is that when there is uh, when there is cutting off of the branching point at below the aspartate semialdehyde step, then there will be shortage of threonine and methionine, which are essential amino acids in the bacteria in the bacterium. So what we will do is that we will provide threonine and the methionine in the medium that is that will be used to grow Corynebacterium glutamicum for lysine production. This will ensure the availability of threonine and methionine uh, amino acids for the normal growth of this bacterium. Now after this, another strategy that we can use to engineer the growth medium is to include some inhibitors of aspartate kinase and the homoserine dehydrogenase, where the cells to be grown are not deficient or not being mutated. There we can include these inhibitors of aspartate kinase and the homoserine dehydrogenase. These inhibitors are these inhibitors bind to these enzymes at allosteric sites, and they relieve or deregulate these enzymes. How do they deregulate the enzymes? They, after binding to the allosteric sites of these enzymes, they reduce the affinity of this enzyme to bind to the feedback inhibitors, for example lysine and threonine. So once these metabolites, these inhibitors, namely aspartate hydroxamate for aspartate kinase and alpha ketobutyrate for homoserine dehydrogenase, when these are bound to the respective allosteric sites, lysine and the threonine will be no longer able to bind to the uh, to these enzymes. Hence, there will be no feedback inhibition induced by lysine and the threonine. Remember, this process is to increase the production of lysine. When we increase the production of lysine, it is bound to uh, induce inhibition of the enzymes. But if we include this inhibitor, then this increasing number of quantity of lysine in the medium will be now not able to bind to the enzyme to inhibit it. So this is another strategy of process engineering. So there are other strains of Corynebacterium glutamicum also, like leucine oxotropes. There, from threonine, there is another branch which also leads to production of leucine amino acid. So if there is a there is one strain which is leucine oxotrope, so which means that it is not able to produce leucine. So the branching point from threonine will be cut off, and there will be no additional problem coming in from uh, the from leucine amino acid. But in this case, if leucine is not produced, then we have to supply leucine or include leucine in the medium for Corynebacterium glutamicum's growth. The pH is 7 and the temperature required is 30 degrees Celsius, and the whole culture should be carried out in aerobic condition. And from all this, the possibility of lysine production is 80 grams of lysine per liter of medium in 27 hours. 27 hours means the whole process of fermentation is being carried out in batch process or batch manner. By using this fermentation strategy, 8 lakh tons of lysine can be produced in industries per year. So here, to sum up the engineering strategy of lysine production in Corynebacterium glutamicum, what I would say is that first of all, a leucine oxotrope was taken. These strategies could be done in isolation as well as in combination. The leucine oxotrope, if these are to be done together or in combination, then let me sum up what will be the actual engineering strategy. Take a leucine oxotrope and mutate it to increase it, it to increase the expression level of home, Lyc and PYC genes. Remember, these genes are uh, important for the production of lysine, and in the wild strain of Corynebacterium glutamicum, these genes are expressed in very low level. So by inducing mutation in these genes, if we are able to increase of course there are reports of increased expression of these genes. So in the leucine oxotrope, we are mutating these genes by to in order to increase their expression level. After that, the cells, the mutant cells, are cultured in a medium containing provided with threonine and methionine, and also inhibitors of aspartate kinase like aspartate hydroxymate, and inhibitors of homoserine dehydrogenase like alpha ketobutyrate. These are provided, and then the pH is maintained at 7 and the temperature is maintained at 30 degrees Celsius, and the process is carried out in aerobic environment. This is the process engineering of lysine production, wherein the upstream engineering involves strain improvement as well as process engineering combined together. These are done separately, uh, um, uh, but still these two, when they are combined, will bring about increased production of lysine in industries. The next example is of citric acid production by aerobic bioprocess. Citric acid is a primary metabolite which is produced in citric acid cycle. This picture shows the complex pathway wherein citric acid is produced and it is converted into different other intermediates, which are very much important in a biological system where there is respiration taking place. If we have to look at the controlling points or the points of regulation of the citric acid or tricarboxylic acid cycle, then what we can do is that citric acid production can be by increased by using a mutant lacking an enzyme of the cycle, any of these enzymes, and by inhibiting the flow of the cycle. If any of the enzymes involved in the cycle is mutated or is not able to carry out its normal function, then there will be accumulation of the intermediate, and subsequently there will be increased accumulation of citric acid, because citric acid is the starting point of this pathway. When any of the downstream enzymes are not able to carry out its normal function, uh, the flow of the intermediates will be stopped, because of which there will be accumulation of citric acid. Now in industries, what kind of engineering they do for production of citric acid is detailed here. In this strategy, the source or the enzyme or the microorganism used is Aspergillus niger. It is the main bacteria fungi which is used for the fungus which is used for the production of citric acid in industries. Now let me go through this first and let me tell you why this strategy is uh, uh, in use. Citric acid production uh, is possible in mixed growth associated, mainly that takes place under nitrogen, and citric acid production is mixed growth associated, that is where the cells are growing uh, and but they are not dividing, their metabolic process is still, yeah, taking place or happening, and that is possible only when there is a limitation of nitrogen and the phosphate. So citric acid production happens when there is nitrogen as well as phosphate limitation or stress, and the growth or the division of cells has stopped. Now for this, what are the medium requirements for high production or production of citric acid? Carbon source required is molasses or sugar solution in the range of 15 to 25%. Then sodium ferrocyanide to reduce iron and manganese contents. High dissolved oxygen concentration, since the process is uh, going to take place in aerobic environment, dissolved oxygen is also required. High sugar concentration, the sugar concentration here 15 to 25% is high, and the pH requirement for citric acid production in the medium is 1.7 or in between 1.7 to 2, and the temperature requirement is 30%. Now why all these requirements are there? One important thing that I would like to highlight in the case of industrial production of citric acid is that this strategy or upstream processing strategy of uh, industrial production of citric acid does not involve any metabolic engineering or any kind of mutation or things like that. What we need is that uh, process engineering. When we engineer the process, that is only when we tinker with the medium components, there will be uh, high production of citric acid without any biotechnological or metabolic engineering approach. So how the components uh, of the medium brings about these uh, high production of citric acid is now to be seen. Let me come first to the low pH which is used for the production of citric acid. How low pH favors or facilitates accumulation of citric acid? Now at low pH, this fungus Aspergillus niger exists in a morphology with small aggregates of short filaments that produce optimum quantity of citric acid. The pH ensures a particular morphology of this fungus which will help in production of high quantity of citric acid. And there is sodium ferrocyanide, which is added to remove iron and the manganese. So how this will help? This will help. Manganese is required by enzymes, many enzymes, and those enzymes are involved in cell function, cell wall synthesis, sporulation, and the secondary metabolite production. When manganese is in short supply in the medium, all these functions will be stopped, which means that you see this citric acid production in the first point. What is given is that it takes place in mixed growth condition, that is, and also when the growth has ceased. So in order to uh, contain or to stop the further growth, what we need is to stop the cell function, to stop cell wall synthesis, to stop sporulation, and to stop secondary metabolite production. And all of this can be achieved by controlling the level of manganese. So sodium ferrocyanide is going to reduce the concentration of manganese, thereby the cells will be now not in a position to divide and carry out other functions which will come in the way of production of citric acid. Phosphofructokinase is one enzyme which will ensure the entry of uh, metabolites like pyruvate or pyruvate, which will uh, convert into acetyl CoA and enters the citric acid cycle. So if the supply of acetyl CoA to enter the citric acid uh, cycle, if the entry of this metabolite is low, then what will happen is that there will be no uh, uh, production of citric acid or reduced production of citric acid. So there should be continuous supply of this metabolite. The pyruvate should be in continuous supply. So in order to ensure continuous supply of pyruvate, phosphofructokinase should be uh, deregulated, or it should be working continuously. So this can be achieved by reducing the concentration of ATP in the cell. Now when there is the presence of iron and manganese, these will ensure uh, two things. One is to reduce the concentration of ATP by not allowing electron transport chain to take place, and also by controlling the degradation of citric acid by isocitrate dehydrogenase, which is magnesium dependent. So now uh, phosphofructokinase uh, in the presence of low pH and high citric acid accumulation will be deregulated, because when citric acid accumulates, it means that electron transport chain is not taking place um, uh, properly. So when electron transport chain does not take place properly, there will be low level of ATP. And there is another uh, okay, let me come to the next point, that is phosphate and nitrogen limitation. When the cell accumulates high quantity of citric acid, this cell will be uh, having very low pH, which is not good for the cell. But remember, the production of citric acid is taking place when these cells have ceased growth, which means that, and also there is limitation of nitrogen and phosphate. When there is limitation of nitrogen and phosphate, there is increased degradation of proteins that leads to increased ammonium ion concentration. Proteins are nitrogenous metabolites. When proteins degrade, ultimately it forms nitrogen compounds such as ammonium ions. So when ammonium ions, which are basic in nature, increase in the cell intracellular environment, the acidity because of the high level of citric acid will be countered or will be offset. That is how this cell will be able to survive in even when there is high level of citric acid. Also, uh, the citric acid inside the cell is not going to be present or accumulated in the cell for quite a long time, because the tricarboxylate transporter in this cell will be activated, and it will send out the citric acid into the medium. So inside this cell, uh, the low acidity is offset by increased concentration of ammonium ions, uh, and the outside the cell, when citric acid comes out, it is not going to affect the cell's normal uh, cell's normal um, working, because when the pH is low, the cell prefers, or the cell is optimized for accumulation of citric acid. High concentration of ammonium ion and glucose. Here, glucose actually represses the uh, synthesis of alpha ketoglutarate, which because glucose inhibits or repress the enzyme that is alpha ketoglutarate dehydrogenase. So this is actually and when glucose inhibits this enzyme, it leads to inhibition of citric acid catabolism. From alpha ketoglutarate, there is a branch which leads to uh, leaking of the citric acid cycle intermediates, and there is not going to be accumulation of citric acid. But when there is high concentration of glucose, that leak is mended, and there is no leakage anymore, and it leads to accumulation of citric acid. Now, about the degradation of citric acid by associated dehydrogenase, I have already mentioned that the short supply of manganese is going to take care of that. And similarly, so when we ensure all these medium components present uh, in the culture environment or in the bioreactor, there will be high industrial high and there will be high production of citric acid. So as we have seen so far now, is that there is increased production of citric acid even without any biotechnological intervention or biotechnological strategy um, to alter or to modify the organism, that is Aspergillus niger. Just um, by tinkering or by playing around with the medium components, we are able to get high level of citric acid production in Aspergillus niger. So this is one strategy where the process engineering is bringing about sufficient level of upstream um, processing. Although upstream processing involves two components, one being strain improvement and the other being process engineering. In this case, there is no strain improvement involved, there is only process engineering. The next example, okay, after this engineering, the production of citric acid happens in a batch. This batch cultivation is required uh, to reduce substrate inhibition and prolong the production phase by 1 or 2 days after the growth cessation. So the bioreactor which is used for citric acid production is of 100 meter cube, that is 1 meter cube is equivalent to 1000 liters, which means 1000 into 100, that is 1 lakh liters of um, um, liter capacity bioreactor is involved. Then here the inoculum should have a particular quantity, and aeration is required in the range of 0.1 to 0.4 volume of air per volume of meter. Temperature is controlled by cooling the bioreactor using coil and uh, cooling jackets. Agitation should be minimum or less, that is in the range of 50 to 100 rpm. High level of agitation will lead to shear damage on molds. Volumetric yield of this whole process cultivation will be 130 kilograms of citric acid per 1000 liters. So in a year there is around 18 lakh metric ton citric acid produced by using this uh, fermentation method. Now citric acid is released into the medium, so the downstream processing of citric acid is achieved by uh, precipitation and crystallization. First, the cells are separated from the medium, then uh, by filtration, and the medium is transferred to a recovery process system, and there separation of citric acid from the liquid happens by way of precipitation. The precipitation is in the form of calcium citrate tetrahydrate. By when calcium hydroxide is added into the medium or the liquid, calcium citrate tetrahydrate precipitate is formed. This precipitate is washed and then it is dissolved in sulfuric acid to yield citric acid and calcium sulfate precipitate. This precipitate and this citric acid is now bleached and crystallized to get anhydrous or monohydrate citric acid. This is how we get citric acid in our labs and for other common uses. Citric acid is an important preservative, an acidulant, and there are many other uses of citric acid, particularly in food industries. The next example is of antibiotic production. The antibiotic in uh, being discussed here is penicillin. Antibiotic penicillin was purified only after 1940s when the Second World War was happening. Penicillin was the first important commercial product produced by an aerobic process submerged fermentation. When penicillin was first made at the end of the Second World War using the fungus Penicillium notatum, its efficiency was just 1 milligram of penicillium per decimeter cube. 1 decimeter cube is equivalent to uh, 1 1 centimeter cube is equivalent to 1 liter. 1 decimeter cube is equivalent to 100 liters. Today, this efficiency has increased to 50 grams from 1 milligram of penicillin. Now it is 50 grams, so there is 50,000 times increase in the efficiency of penicillium strain by using Penicillium chrysogenum. Earlier it was Penicillium notatum, but now it is Penicillium chrysogenum. But still, the achievement is remarkable. That is 50,000 times increase in the efficiency. But still, the requirement of penicillin is such that we need to improve the yield further. So how do we increase the yield further, and how we have increased the 50,000 times increase in penicillin production? Now if we look at the biosynthetic pathway of penicillin in fungi, what we see is that it starts with a condensation of L-amino adipic acid, L-cysteine and L-valine in the presence of one enzyme called ACV synthase. It is encoded by a gene called PCAB. Now, when these three amino acids combine, there is formation of amino adipic cysteinate valine, or it is AC commonly called ACV. So from this intermediate, this is the first intermediate of the penicillin biosynthetic pathway. This intermediate is converted into isopenicillin in the presence of isopenicillin N synthase. This step requires ascorbic acid, iron and oxygen. Now from this isopenicillic acid in the presence of isopenicillin N-acyl transferase, 6-amino penicillinic acid is formed, which is the precursor of all, or which is the core or the functional portion of all penicillin related antibiotics. 6-amino penicillinic acid is converted into penicillin G in the presence of isopenicillin N-acyl transferase. This gene is encoded by PCBC, and penicillin G can be also produced from phenylacetic acid after converting it into a ligated intermediate in the presence of phenylacetyl-CoA ligase. This ligated intermediate, which is acyl-CoA phenylacetic acid, acid CoA, which is converted into penicillin G in the presence of acyl CoA 6-amino penicillinic acid acyl transferase in the which is encoded by PEN-DE gene. This is the pathway of penicillin biosynthesis in fungi. Now, in order to improve this pathway, the efficiency of this pathway, we need to improve the strain, the fungus in hand. There are two ways of uh, bringing about mutation in the strain. One is physical and the other is chemical. By physical means of mutagenesis, we mean exposure to UV radiation and such exercises, but it was not found to increase the mutation, I mean to the increase the production of penicillin to a great extent. So chemical mutagenesis was considered. By combining this physical and chemical mutagenesis, one could get only 60% reduction in one could get only six-fold increase in antibiotic production, but the the mutagenesis also reduce the growth of the cell by growth of the cell by 60%. So this process is not good in the sense that it is inhibiting the growth of the organism, and because of that, the efficiency, the increase in the efficiency, that is sixfold, is not considered quite significant. So we need to find out other strategies of increasing the production of penicillin. The other possible strategies are recombinant DNA technology, and for recombinant DNA technology, what we can do is increasing gene copy number in the organism or strain which is to be used for penicillin production. Here in Penicillium chrysogenum, people have tried to increase the gene copy number of the biosynthetic genes PCBAB, PCBC, PENDE. You can go move to the previous slide to see uh, what these genes, which proteins these genes encode. But by increasing the copy number of these genes, what people could achieve was just 40% increase in the penicillin production. But in the case of Aspergillus nodulans, another fungus, what people could achieve was 40% was 30-fold increase in the production of penicillin just by overexpressing one gene, PCBC. This PCBC gene is involved in the production of or expression of isopenicillin N acyltransferase enzyme. So in this biosynthetic pathway, particularly in Aspergillus nodulans, what is apparent is that overexpression of PCBC, isopenicillin N acyltransferase enzyme, is the key enzyme, and by overexpressing this enzyme, we can bring about 30-fold increase in the penicillin production. So in the case of penicillin, the strategy to increase penicillin production is to overexpress a key enzyme of the biosynthetic pathway. Unlike in the previous example of primary metabolite production, that is citric acid production, and lysine production, in the case of penicillin, which is a secondary metabolite, the production of uh, penicillin is brought about by overexpression of a key gene. That was about strain improvement. Now let us come to the process improvement of fungi of a fungus for the production of penicillin. So industrially, Penicillium chrysogenum is used for the production of penicillin. So the strain improvement which we have just mentioned here in the case of Aspergillus nidulans, by overexpression of uh, PCBC, which encodes a isopenicillin and acyl transferase, can be also done in Penicillium chrysogenum. So by engineering Penicillium chrysogenum, we will use it for the production of antibiotic, particularly penicillin. So in the when the process engineering happens, penicillin is uh, cultured in a medium which has lactose, other sugars, and a source of nitrogen. And the source of nitrogen in this case is a yeast extract. The fermentation process is in batch, and a fed batch process is normally used to prolong the stationary period and so increase production. So in the case of citric acid production, the requirement was the phase wherein the cells stop um, growing, and here in the case of penicillin production, the requirement is also when the cells is the lack of growth of cells, because penicillin is a secondary metabolite unlike citric acid, which is a primary metabolite. In the case of penicillin, which is a secondary metabolite, secondary metabolites are ideally produced only when these cells are in their stationary phase or death phase, just before the death phase. So in the case of citric acid production, this phase uh, wherein the growth was limited, and the limitation of phosphate and the nitrogen was ensured. All these conditions led these cells to uh, accumulate citric acid. But in the case of penicillin production, there is absolute requirement of these cells to be in stationary phase. Here in this slide, there is a flow chart of the fermentation process for penicillin production. Mediums are now put into the fermenter, and the starter culture is added. After that, there is 10 times increase in the starter culture in 6 days, and then remove 30% of the culture and add 30% fresh medium in order to um, uh, increase the exponential phase. Then the cells are separated and the cells are taken out for other uses, whereas the medium or the supernatant is collected, and it is used for extraction of penicillin, which is the downstream processing. In the downstream processing, the filtrate is mixed with butyl acetate, and then potassium ions are added to precipitate the penicillin in its salt form, potassium salt form, and then the precipitate is washed, filtered, and dried, and we get 99.5% pure penicillin, which is further used for chemical and enzymatic modification to make new derivatives or antibiotics. So the downstream processing of penicillin is quite simple. It is also very easy, since the uh, antibiotic is secreted into the medium. There is no need to kill the cells to extract the antibiotic, because it is already present in the medium, not in this cell. Okay, in this slide there is the conversion pathways, or the possible modifications of 6-amino penicillinic acid to different potent antibiotics. So all these modifications are done chemically, or maybe enzymatically, but these are done in vitro, not inside the cell. The next example of primary metabolite production in microorganisms is production of grain fluorescent protein. Grain fluorescent protein is a commonly used marker, or reporter in biotechnological studies in labs. So it is, from the name itself, it is understood that the protein is a, a fluoresce, and the protein fluoresce um, green color when it is exposed to certain excitation wavelength. So how we produce green fluorescent protein which is very much required in research labs and other uh, studies. For this, the microorganism used is E. coli. In this bacterium, we insert this is the strain improvement part. The strain improvement involves insertion of T7 RNA polymerase gene in beta galactosidase gene of lac operon. In the E. coli gene, in lack of peron, the one structural gene beta galactosidase uh, is present, that is now being replaced by T7 RNA polymerase for the purpose of production of green fluorescent protein. How this RNA polymerase will help in producing green fluorescent protein? When we mobilize a plasmid carrying green fluorescent protein gene construct, the plasmid, which is already present in the leg operon of the genomic DNA of E. coli, there is increased replication or expression of there is increased expression of the gene which is present in the uh, plasmid, wherein this gene has been constructed in a particular fashion. So the RNA polymerase will help in better expression of the gene. In order to increase the expression, what we normally do is to uh, increase the production of RNA polymerase protein, that is done with the help of induction of lac promoter by isopropyl beta D1 thiogalactopyranoside, or simply IPTG. IPTG induction is required to increase the expression of RNA polymerase. When there is increased RNA, it is actually required to increase the expression of lac operon, particularly beta galactosidase gene. But in place of beta galactosidase gene, there is T7 RNA polymerase. So when we use IPTG induction method, there will be increased expression of T7 RNA polymerase, and this in turn will lead to expression of GFP protein. So this is the biotechnological strategy we will use for strain improvement for the production of green fluorescent protein. Now in the process engineering part, agitation rate is 206 rpm, temperature requirement is 31 degrees Celsius, pH requirement is 6. Then IPTG induction is done when the optical density of the culture is 1.04. GFP production is done when the GFP production is at the rate of 0.241 grams per liter, which is about 9-fold higher than the unoptimized conditions. And the whole culture is the whole cultivation process is done in batch or fed batch. For downstream processing of proteins, there is anion exchange chromatography and size exclusion chromatography. Now, in the strain improvement part, there is another method which is alternatively used for green fluorescent protein production, that is temperature sensitive promoters from lambda phase could be used to induce the promoter. Instead of IPTG induction, temperature sensitive promoters can be used, and so at 42 degrees Celsius the induction takes place instead of IPTG treatment. When we simply raise the temperature to 42 degrees Celsius, there will be induction happening. So this is another way of inducing T7 RNA polymerase for production for expression of green fluorescent protein. The next example that we will see is of biosurfactant, and in biosurfactant, alpha amylase is one enzyme which is used as an important component of in detergents. So it's particularly its use is as a bio as a protease. Now alpha it is not a protease, it is a carbohydrate degrading enzyme. So in detergents, alpha amylase is an important component. So now when we talk about the production of alpha amylase, we need to understand why there is requirement of more production of alpha amylase. Alpha amylases are starch degrading enzymes, as I said, carbohydrate degrading enzyme that catalyze hydrolysis of internal alpha 1,4 O glycosidic bonds in polysaccharides with the retention of alpha anomeric configuration in the products. It has a wide range of application, right from food, fermentation, textile, paper, detergent, pharmaceutical industries. There are also new applications being proposed in clinical, medicinal, and analytical chemistry. Alpha amylase enzyme or the protein is secreted into extracellular matrix or environment by various bacteria. Industrial production of alpha amylases is usually done from Bacillus species of bacteria. Now for a protein, there are, unlike that of green fluorescent protein, for other proteins which are particularly extracellular proteins, extracellular enzymes, the strategies for genetic improvement are, some of the strategies are listed out here. The first strategy is to enhance the expression of the gene encoding extracellular enzyme through amplification of gene copy number. The gene copy number is increased, and then thereby we get increased expression of the enzyme, that is extracellular enzyme. Also, along with this, there is codon optimization or strong promoters, which are um, which are able to supplement the uh, expression. The second strategy is enhancement of secretion of modulation of signal peptides. When proteins are involved, and some proteins which are to be localized to a particular site, we need signal peptides. So if we enhance the secretion uh, by modulating the signal peptides, if the signal peptides are uh, directing the protein to go out of the cell, then there will be no accumulation of the protein, and when there is no accumulation of the protein, the cell will not be under stress to accommodate the increasing production of the protein. So by modulating the signal peptide, there can be increased or enhanced secretion of the protein. So this will ensure continuous production of the protein. Of course, along with this, there are some other adjustments to be done, like fusion to a heterologous signal peptide for efficient targeting to the translocase, increasing the gene copy number of signal peptidase genes, or deregulation or co-expression of chaperone encoding genes to make efficient protein folding. The third strategy is to redesign the capacity of the secretion machinery. The second example is also related to secretion of the protein. The third example again emphasizes that there is a need to redesign the capacity of the secretion machinery by targeting by targeted deletion of genes encoding non-beneficial extracellular enzymes to avoid or to limit the competition in secretion of extracellular proteins. So by removing or by somehow limiting the requirement or need for excretion of non-beneficial extracellular proteins, we will be able to achieve efficient secretion of our extracellular protein. The fourth strategy is to prevent degradation of extracellular enzymes using protease deficient strains. Certain strains are good in production of proteases, which will reduce some extracellular proteins. So this is done in order to protect itself from invading extracellular proteins, which will degrade the external parts of the cell. But we can, when we use one strain for production of extracellular proteins, we can uh, do away with this mechanism, because there is no external threat going to come to this strain of our choice. So we can prevent degradation uh, by using protease deficient strains. So after seeing this background, now we will talk about production or process engineering of alpha amylase. The strain used for alpha amylase production is Bacillus licheniformis. This strain is selected because naturally this strain produces a good quantity of alpha amylase. It may not be sufficient for industrial scale production or the requirement of alpha amylase or the demand of alpha amylase to be met, but still it is important because of its high level of accumulation of alpha amylase. The strain which is industrially used now has undergone a significant biotechnological uh, modification. Now, the modification which has taken place is that this strain now is called CBBD302, in which there is single or double mutation or double knockout of type 1 restriction modification system in this microorganism, in this in this bacterium, which means that the transformation efficiency of this bacterium is very low because of the presence of type 1 restriction modification system. So when we delete this whole machinery of type 1 restriction modification, the cells become more readily amenable to transformation, and by uh, by increasing the transformation efficiency, we will be able to bring about uh, greater or higher production of alpha amylase in this strain. Now for this, one high AMIL plasmid is used to modify this bacillus like caniformis, which is now free of type 1 restriction modification system. When this plasmid is mobilized into this strain, uh, there is possibility of increased production of um, alpha amylase. Now this organism, this particular bacterium, is already good in production of alpha amylase. Apart from that, we are mobilizing a plasmid which is carrying alpha amylase gene. So in addition to what is naturally produced in the bacterium, we are also going to get some production from the plasmid that we are mobilizing into the cell. For the process engineering, this microorganism is uh, in a catabolite repression condition when there is sugars such as glucose are used for growing the cells. So now we have we use lactose instead of glucose. Glucose is a monomer, whereas lactose is a disaccharide. So a disaccharide is used instead of a monosaccharide to avoid catabolite repression. Apart from this, this is a source of carbon or sugar. Now the other medium requirements like protein source from soybean meal and cottonseed meal are used. Also corn stable liquor is used as a source of uh, protein or nitrogen, and the ammonium sulfate is also used as a source of inorganic source of nitrogen. The pH required is 6, and from this whole process of cultivation, we will be this is done in 15 ml capacity, 15 liters quantity in batch fermentation for 78 hours. The RPM requirement is 220. Inoculum required is 5% volume on volume basis. The temperature required is 42 degrees Celsius, and the yield we get from this whole process of cultivation is 15.6 milligrams of alpha amylase per ml broth medium. Downstream processing of alpha amylase is almost the same as that of uh, GFP protein, because GFP, green fluorescent protein, is also a protein and alpha amylase is also a protein. So most of the proteins can be separated by using the same method of separation. Now in order for this alpha amylase to be processed or to be purified, first the ammonium sulfate precipitation is carried out, and then the the precipitate is collected and dissolved in glycine or glycine buffer of pH 9, and it is dialyzed overnight against the same buffer. After dialysis, the enzyme solution is filtered using a membrane. Then the enzyme solution is purified to get purified through anion exchange column to get different level of purity for different end uses. Okay, we will stop here with this for today. I will continue this in the next lecture.